Another technical session. My name is Stefan Winter. I'm from the Leibniz Institute in Germany. I'm dealing with plant diseases in general and tropical diseases in specific. And I will guide you through this afternoon. So we have 5 presentations, and I think we should, if you all adhere to our timing, we still have a small segment for discussions. And I would like to here with ask our first presenter, and this will be Johannes Kopsen, to when and where to warn farmers. So you're most welcome to your presentation, and I would like to ask you to speak to the microphone because otherwise streaming is difficult.
Thank you very much. Yeah. Yeah. All right. Thank you very much. Yeah, I'm Johannes Kopsen. I'm from the University of Bonn. Part of this work was also done in collaboration with the University of Wageningen as a guest stay there. And this presentation is about when and where to warn farmers. And we analyzed the value of the spatiotemporal pest risk prediction for pest monitoring decisions. And this is about smartphone-based pest risk warning system. And the question we naturally ask for this is when and where to warn farmers. And kind of the obvious or naive answer would be, yeah, when the pest risk is high, right? I mean, it sounds logically, but actually, if the pest risk is really high, farmers will probably be aware of it, and then it will be probably a very common disease. And people will already know what to do. People will have their treatment ready, and it's not really news for them. So we think it's actually quite more valuable if we warn farmers not when the pest risk is necessarily high, but when it's changing the farmers' management decisions. So in the end, a pest risk warning system is only really valuable if it has actionable consequences for the farmers. And therefore we developed a decision model with 3 decision options, namely treatment. This could be application of, for example, an insecticide, and then monitoring. And by monitoring, we mean going to the field and looking at the crops and checking if there's a pest infestation. And then thirdly, obviously, inaction, doing nothing. For all of these decision options, we developed economic utility functions, calculated the overall costs. And we are particularly interested in the value of information for the monitoring. And this is kind of the expected monetary benefit of going to the field and checking the field for pests. So usually, actually, yeah, nobody really needs this pest risk monitoring system, because we could go every minute to the field and check every minute the field for a pest infestation. Then we wouldn't need a model for this. But this obviously is, yeah, very expensive in terms of time and resources. We can't check there so often, and we therefore want to know what is actually the value of going there. What do we actually benefit from checking the pest for infestations? And this question can be answered once for the individual, because obviously it has a benefit for me as a farmer when I go to the field and check for pest infestations. But also if I use then this information and feed it back into the model, use it to inform the model so other farmers can be warned, then it has also a value for the community. So overall, my neighbors in the future, maybe they get an alert and then they will be warned because I went to the field and I found pest infestations there. So let's have a look at the pest risk prediction model itself. Yeah, therefore we use this well-known disease triangle. You probably all know this. It depends, the pest infestation depends on the susceptible host, and this we expect the users of the model to know. The users know which crops they grow. Then also a conducive environment is necessary, and this we infer from environmental data, mainly weather data. And the pathogen presence data we infer from observations from the spatio-temporal neighborhood, so from other farmers nearby in the near past. The data source were observation data from the Plantix Android app. This is actually the most used farmers app in the world. It is developed by a company that was originally a spin-off of Hanover University, and they have this unique crowdsourced data set consisting of millions and millions of data points, each built of location, timestamp, and the detected pests. And let's have a quick look at the, we can't really see this here, but have a quick look at the architecture of the model. The weather input is first, it's a kind of a combined machine learning like model, but it's not your average neural network. But first the weather data is encoded into latent features using a neural network component. Thank you very much. And these latent features are then fed into a Gaussian process layer. This is kind of like, maybe you know, kriging for spatial interpolation. It's kind of the same basic thing. And then it is combined with the spatial information and also the timestamp. And altogether, this then combined to calculate a similarity metric between points for this kind of interpolation, also extrapolation thing with this Gaussian process. I won't go into too much detail here, but in the end we get a pest probability. So that is kind of our pest risk measure. And as a case study, we used tutor absoluta. I really like this letter name, so I will, from now on, I will say tutor absoluta all the time because I like it so much. It's also called tomato leaf miner, and it affects multiple crops, but we focused on tomato. It is in India, it is an invasive pest because it originally comes from South America. And usually farmers respond to it by really heavy schedule based insecticide use. So usually every 10 days, they will go to the field and apply some insecticide. And as a case study area, we use this Indian state Andhra Pradesh. Let's have a quick look at the results. So here we can see the predicted tutor absoluta risk for 3 exemplary days. And for example, we can see at the 1st of June, there are these 3 kind of hotspots. And these are because in the past there were some tutor absoluta observations, and also the environmental conditions, like the weather conditions in these spots, were very favorable for the development of tutor absoluta. So we have a higher risk there. We can also then map the value of information for monitoring. So the more purple it is, the more value we will get from going to the field and looking at the infestations. And this we can then use to create these maps with the recommended decision options. So in the blue areas, actually inaction is the favorable condition according to the model, the favorable management action. Monitoring is favorable in the orange places, and only in the magenta ones, spraying is considered optimal. And that I find interesting because that is what farmers actually in the entire state basically do as a treatment for tutor absoluta. But according to the model, it's only favorable in certain places. And then we did an agent-based simulation in order to find out what would be kind of the aggregated effect if every farmer in the entire state would use the model and act accordingly. And here we can see the costs from different strategies, and our model strategy is the 4th one. And as we can see, lower is better, right? These are costs. So the strategy actually has some impact in the simulation, some positive effects. It's actually better than the farmer's practice of calendar spraying. But we can also see these positive benefits we can only really observe if the model is updated by new observations from the users fed back into the model and used to inform the model and used to warn other farmers. So if we have no updating in the model, as we can see in the lowest one, then we don't really see these effects. To sum this up, we can say that the costs could be reduced in the simulation by about 10%. And the pesticide use could even be reduced by 60% from using the model recommendation. And for the value of monitoring, we can see that one round of crop monitoring, when it was recommended, saved an average 37 rupees. This is like 40 euro cents for the individual farmer. And what I find interesting is that for the community, it was even more. So the value they generated for the community by informing their neighbors, warning their neighbors, and using these observations they produced was even higher. And this maybe these couple of euro cents doesn't sound much, but if we add this up over the entire season, then it is really multiple times daily wage for a farmer in India, in Andhra Pradesh. So it's really a considerable amount of money in the end. And yeah, by that I want to thank you. Yeah, we submitted also a full paper, and also the code for this will be open source soon. And yeah, thank you very much.
Thank you very much indeed for this nice presentation. And I can allow 2-3 questions on this. Which gives me the first one. So how do you deal with uncertainty? So when you make a prediction, on what scale do you do this? So tuta absoluta responds as an insect on a very small scale. So when you make a model over a large area, how sensitive are you in sort of hitting your target? Or you deal with the uncertainty that you provide a prediction model over a larger scale, geographical scale?
Yeah, thanks a lot for the question. So uncertainty plays a very large role in this. Everything basically is here uncertain. In the end, I did not really report in this presentation too much on the standard deviation and so on. I apologize for this, but in the end, everything was uncertain. Also, these risk, everything like the interpolation. This is kind of the beauty of the Gaussian process approach, that it automatically gives you also the uncertainty from this interpolation kind of thing. And as a spatial unit, we used the grid that was also the environmental data, the weather data. It was 11 kilometers, I think, for a grid cell. And yeah, then in areas where we had a lot of data, like the standard deviation would be lower, and in areas where we had little data, the standard deviation would be higher, and the information would be more uncertain. Yeah.
Thank you for your nice presentation. How would your model actually deal with current and tests? So Pest, where the probability of entry is very low, but in case of an outbreak, the impact would be very severe.
Thank you. Yeah. This is very interesting. For this particular project, we only looked at tuta absoluta, which is kind of common, even though it is originally like an invasive pest. So it is not really fulfills these criteria. But I think it really depends a lot on the disease, what kind of effects we can see here. And for some diseases we expect a lot more like spatio-temporal autocorrelation, like really these diffusion kind of things, really these outbreak patterns. And for some pests or diseases, maybe if we even look at fungi or something, then we expect a lot more dependence on the environmental factors. And the predictors will be different, but the model will be kind of the same. I mean, this can deal with both worlds, so to say, the diffusion or autocorrelation kind of things, and also the covariate information.
One last.
Thank you for your very beautiful presentation. I noticed the temporal dimension is very good. You checked the February, then July, then September. So I don't know if you were able to look at the manual window for Pest is very fast. After effective reduction of pesticide application, it has to be like near real-time monitoring. Have you compared the plots here and the last reduction based on the new?
Thank you. Yeah, the temporal resolution of this project was actually 1 day. So these are just 3 exemplary days. This is not one time step. This is like many time steps. Yeah, and yeah, one day we had like weather data in the resolution of 1 day. We could even have a higher resolution because I think the observation data we have is really an exact timestamp. Even the time of day, we could do it on a millisecond based thing, but I think that would be overkill. But yeah, one day is here the scale.
Thank you very much for your last presentation, for you contributing to those questions, and I will get up. The applause to our next speaker is Kai Behne, who speaks about invasive plants. Here is him. Yeah.
Good afternoon, everyone. My name is Kai Behne, also from the University of Bonn. And I have the honor now to give a speech on invasive plant species drive land use changes in East Africa. And this is the work of a German East African team. So it's not only my work, I'm just part of the team, but I'm presenting on behalf of the group. So where is this area we are looking at? This is in Kenya and in Baringo County in Kenya, and specifically and around the Lake Baringo on the southwest of the Lake Baringo, in a semi-arid climate. And in this area, the traditional land use is pastoralism with rangelands. But in this area, there was in the 1980s a plant, shrub or small tree introduced called Prosopis juliflora. And the idea was to combat desertification and improve the livelihoods of the people, for example, to have a tree which can be used for firewood. And this tree is an evergreen, has thorns, and it's coming from America. It's a phreatophyte with deep roots accessing to the groundwater. Even when the plants are still very small, it can go very deep. And it's a nitrogen-fixing legume. So there are several factors which make it, which are traits which makes it very suitable for this environment, and the plant well established there. But after some time, it became an invasive species there and became a big problem. And one of the reasons why this happened is that there was droughts in the region and farmers used more and more goats instead of cattle. And the goats were eating the pods of the Prosopis and spreading the seeds all over the place. And then in the 2000s and also in the 2010s later, there was a massive spread of this species. One of the problems associated with the plant is that it has allelopathic effects on other plants, for example, preventing germination of C4 grasses by substances exudated by the roots or from the leaf litter. And this leads to the fact that the plant indeed has some positive effects on livelihoods, for example, as intended with the firewood, but it has also severe effects on pastoralism with a severe decline on rangeland productivity and forage supply in the region, and also by the thorny shrubs creating physical barriers for the animals to move and also to access water points, because it tendentially grows in wetter areas around the lake, so it was difficult for the animals to access the lake in the dry season. And this caused land use changes because in many parts, the traditional land use with the rangeland was no longer possible. So there were initiatives from different stakeholders to increase the infrastructure, to give market access to the farmers, and contracts for farming, to make initiatives to cut off the invasive species from the land and have new possibilities for agriculture. And that led to a shift to crop farming from cattle to corn. And now it's the question, is now the problem solved with these land use changes? And as I still have some minutes to speak, of course not, because after some time of success, the species Prosopis also increased in the croplands, particularly in the rain-fed croplands. But there was also another invasive species called Parthenium. Parthenium is an Asteraceae and it's an annual forb, and it's also originating from tropical America. And it's considered noxious weeds in many parts of Australia, Asia, and recently, in the recent years, increasingly in Africa, in many countries in Africa. And if you look at scientific publications regarding Parthenium from the African continent, it's really skyrocketing, because a few years ago it was not a big problem, and now it's increasingly a problem in many regions. And also, this plant has allelopathic effects on other plants, but also it has negative health impacts both on humans but also on livestock. So even while it produces large amounts of biomass, it cannot be used as forage because it's toxic, and also has effects on the biodiversity in the region. So that makes it a huge problem, which doesn't have any benefits, unlike the Prosopis tree we had before. So what's happening in the region now? It's agricultural intensification, and this is data from a recent master thesis from our group. And it says that in the last 5 years, there's a huge increase in mechanization, mechanical tillage, due to contract farming and the availability of machinery and inputs. And we assume that this mechanical tillage can be a vector for the species, that, like the seeds of Parthenium, are transported from one field to the other, but also that there's a lot of disturbance in the soil which favors annual species, and this is one of the reasons why this spreads so much. A second thing is there's a strong increase of mineral fertilizer, which is also due to the availability, but also farmers were telling us that they are reluctant to use manure because they are fearing to spread the invasive species around there. And also the chemical weed control has strongly increased. And this is, of course, not only due to the invasive species, but it certainly plays a role with the problem of the invasive species. So we see here, now there was the initial phase, decline of forage, and then forage productivity in the rangelands, then a shift to crop farming, and then again, the new problem with the Parthenium and Prosopis arising. So what is now the question for the future is, how can resilient land systems look like that take into account these species? And one difficult point is here that continuous cropping, like for example in irrigated agriculture, reduces the risk for Prosopis but favors Parthenium. So what works against the one invasive species can favor the other invasive species. So there's a need for integration of competitive species, for example, and there's one thing which is called forage seed production, that there's a grass species produced in the area for the seeds and that to sell them elsewhere, and the species is called Cencus ciliaris, and this is very competitive against Parthenium. So in these fields where they produce the seeds for this grass, there's virtually no Parthenium. And this could be an idea for the future to include that also for forage, but of course farmers need to be willing to grow forage. And this could be a challenge also for the future. And another thing is that healthy and regenerated rangelands can also help against both invasive species, because where there's a dense vegetation cover, there's less entry points for the invasive Parthenium to increase. And other things is prevention of further spread, like cleaning of machinery and treatment of manure, and also for the pods, because when the pods are fed to animals, they are milled before, and like this, the plants cannot spread. But when they are eaten by free roaming goats, of course, this is not the case. And so there's a need for control options. And while for Prosopis, for the shrub or for the woody small tree, there are a lot of options also from other regions in Kenya and in East Africa, and they work. But the big challenge will be to have management options for the Parthenium to reduce that spread. And with that, I would like to thank you for your attention. And just the last word, so this was a work of several people. So they did farmers interviews, field assessment, and literature review, and so on. Thank you very much.
Thank you very much for this nice and very, very important contribution. Is open for discussions.
Excellent presentation. It's an area I work in, so I know it well. There's a third invasive species, prickly pear.
Yeah.
Did that come up in your farm interviews?
Our work is not focusing on that, but I know that also I've seen it also in the region. But I cannot say, but I know that they use biological control against that now in Kenya. But sorry, I cannot give you more things to that.
So indeed, Parthenium is really a disaster thing, but it also produces an enormous amount of seeds. So, and these seeds fly. So, I mean, we have seen that in the Democratic Republic of Congo, where it has been introduced these contaminated seeds from others, that in a very short time, it basically was the predominant question. But I mean, here, I mean, the other thing is, probably you have not seen this, but it also can carry viruses. And they fly basically along with the spread with the Parthenium. So is there any option that we can do as a cultural control? For example, can you grow a perennial crop to push it backwards, like cassava that stays there for more than a year? Or is there a cultural control for this? Or do we have to just live with this? Yeah. Because here you basically say, well, let's grow grasses, but this is one option which is nice, but not for every farmer who just says, okay, let me give up my crop production for grasses.
I think with the perennial crops, that's a good point, because what's mostly grown in the area is maize and green grams, which is both annual species. And so I do think that trying to get other permanent cropping systems could help, at least for the Parthenium. Don't know about Prosopis, because one thing that works against the Prosopis is a regular disturbance of the soil, but this favors Parthenium. So I think that's a big dilemma of the thing, that one species is favored by what helps against the other. So, but I think that there need to be options to explore perennial crops.
Thank you very much. Any more? Madam?
Thank you. It's just a quick one. All of the controls you mentioned, integration of Seneca ciliaris. I want to ask, is that native to the area?
So, yes. So the Seneca ciliaris, also called African foxtail grass, and it's known also from Asia that it's competitive against Parthenium. But there's a warning that this plant can be invasive itself if you grow it elsewhere. But this is not so much the case in Africa, where it's actually native. Yeah.
Thank you very much for this wonderful presentation. Here we come to our next speaker. Let the clown begin. Well, we have dealt with modeling the spread of insects. We have dealt with invasive plants. We are now dealing with invasive or that might be invasive new pathogens for prediction of pathogen spread. I'd like to introduce Samashit, who will enlighten us for pests that are somewhere else in the world that might threaten the African continent once introduced. All along, with our invasion theories, new pests and diseases are to prevent, and I will give you the floor.
Thank you so much. Good afternoon, everyone. So today I would like to speak about the cassava wishbone disease, which is now an outbreak in Southeast Asia. So I start with the cassava. I think most of you know what is cassava and how a happy cassava looks like. So this is a happy cassava, no pest, no disease. However, in our department also we deal with a lot of disease and pest, and this is the cassava mosaic disease, cassava brown streak disease. This is mostly what we worked on. So this is viruses in Central East Africa, cassava wishbone disease in Southeast Asia, cassava frog skin is a cocktail of viruses and phytoplasma in South America, and the cassava root necrosis disease. So it's in West Africa and East Africa. So before I dive in the cassava wishbone disease, I would like to give you a bit overview about what we are doing in DSMZ Germany. So here we are, Germany, where cassava is not grown, it's not regulated. However, we have the biggest farm outside Africa and South America, because we work in the center where we can receive the germplasm and we can exchange cassava with our partner. As you see, we have a partner in South America, we have a partner in Africa, a lot of partner in Africa, we do here, a research center, and also in Asia too. So what we do index discover of pathogen, we investigate the identity, origin, diversity of cassava viruses, and we look on a causal agent when new cassava disease appear. Like cassava frog skin disease is not resolved yet, the cassava wishbone, I will tell you later how we resolve it. And we also deal with cleaning infected material. So when someone tells us this is a cassava variety, which is very good, and we need it in our breeding, but it is infected, can you clean it for us? So we do cleaning. So we do also cleaning for cassava. This is the most big part in our research dealing with the cassava brown streak disease and cassava mosaic disease. Identify resistance and try to look what is the resistance from the plant side and also from the virus side. So now I go to cassava wishbone disease, and I have to introduce the team that we are working with them in SIAD. Dr. Jonathan Nubi is the leader of the cassava program in Southeast East Asia and also in SIAD Colombia. Dr. Wilma is the lead group on crop production in Colombia, and he did most of the job. Dr. Juan in East Africa, and the team here, the mycologists and the bioinformatics behind this work. So cassava wishbone disease in Southeast Asia. So all the time was referring to this disease as a phytoplasma disease, and nobody was pointing to fungal until recently. So as you see here, this is how cassava wishbone disease looks like: proliferation, a lot of sprouting, and necrosis. So lately, this disease was connected to the fungus Ceratoplasmidium therapomum. And our part was to look on the localization pattern of this fungus, try to see the tissue invasion, and look for the effector protein. So we have a highly advanced method to look how the fungus localized in the plant. And for the tissue invasion, we looked on pathogenetic protein, and we look on the endogenous protein to look for the localization. And we use the effector protein also to look on the cytoplasmic and apoplasmic localization. So what is really significant about this wishbone disease is make necrosis in the vascular system. And as you see here, this is how cassava stem look like, and this is how you do it when you do a vertical cut. This was similar to a published disease in cacao, the vascular disease of cacao, as you see here. And also it has the similar things. It has a vascular system that is destroyed, so by the necrosis. So mostly when we speak about vascular system destroyed, we speak about plant cells, which is phloem or xylem. So now we want to dive deeper inside the cell and see how they look like. So this is a cassava cross sections. And what is unique about cassava that it has two phloem. It has internal phloem and it has external phloem. So it's not normal that the plant has two phloem, but cassava is one of these plants that has two phloem, and that's also brought the things to that cassava is drought-tolerant plant. It can also handle a lot of drought. So the internal phloem, the plant uses it for transferring the sugar down, and while the external phloem transfer the sugar up, and then you have a regular up-down phloem. So this is the xylem. So, and this is how healthy plants look like. So you see healthy cassava on upper panel there, where the xylem poles are really circular and they are healthy in good shape. While when it comes to the infected witch broom there, you see that these poles start to be a bit dense, and also they don't have the regular shape of the xylem. And here tell us that there's stress happening in the vascular system. So we use this in situ localization technique. It's called RNA scope in situ localization. And we adapt this from the human science. They use it in neuroscience, and it was not widely used in plant pathogens. We use it for the cassava brown streak disease when it comes to the resistant, and we thought we give it a try with the fungus as well. It has a highly specific probe that you use it based on what you want to detect in the fungus. And the sensitivity of this technique is really high because it can go from one dot, so one dot is the fungus here, to a cluster, as you see in the other part. So even one dot, you can, even if the lower, the fungus is not really invasive inside the tissue, you can see it. So this is really highly sensitive technique. So we come then, we say what we have to use as a probe for this technique. And we said, as I said, we decided to use first pathogenicity protein and endogenous protein. The first one here, it's the kinase protein, and it's called, belong to the group of pathogenicity protein because it regulates fungal growth and sporulations, and it also plays a role in virulence. So this is a healthy cassava, as you see in the lower panel there. Compared to infected one, you see a red plaster or a red dots, and this is reflected where the fungus is. So you see it clearly, it's in the xylem, as I said, because this fungus is mostly moving in the xylem. So we want more, and we check in the endogenous protein as related to energy and ATP, and here you see also that mostly the signal was in the xylem as well. So it's reflected by red color. So what we comes to conclusion here, that based on the signal that we see and where the signal is located in the tissue, that it's mostly in the xylem, and this is where we found also the fungus. So this was also aligned with what was published on the cacao with the vascular disease of cacao. So we continue and say, yeah, how is it with the whole plant then? If we find it only where the symptoms is, or is this fungus all, we can find it in all plants. So we check different sections, and I told you this highly sensitive technique, so it will tell us if it's up or not up. And as you see here, the section was clearly clean. So this gives us indication that there's an uncontinuous detection of this ceratopasidium through the whole plants. So the idea comes after that, if it's a case, how we can follow. So we went to the field and we take a plant, a whole plant where it shows symptoms, and this is how the symptoms look like on the cassava. And then we make a section from the whole plant, from the symptomatic part up, and also from the leaves that show symptoms and from the leaves that doesn't show symptoms. And what you see here, the symptoms was morely localized also with where the signal is. And the more you go up, there's no signal. So in this case, also give us a message that this pathogen is localized pathogen. So it's not systemic, so it can't move through the whole plant. And also, it gives us an indication why sometimes when you grow infected Cassava is propagated vegetatively, yeah? So when the farmer takes this and try to plant it, not all the plant will get infected. And this is also explained why that. So we have an explanation for this. So now then we move on the effector protein, and the effector protein are involved in the virulence and suppression of plant infants. And this comes from the fungus itself. And based on the transcriptomic data and proteomic data, we have identified 244 effector protein from this fungus. 196 were apoplastic and 25 were cytoplasmic. So we took these genes and we say, now we want also to see where these genes are localized in the cassava tissue. So the first one is PEP4, and as you see also reflected by red dot, it suppresses the plant immunity, and you see it mostly in the xylem as well. And it's apoplastics. So we went more and to check the other also effector protein, and you check stem and leaf. And as you see here is apoplastic and cytoplasmic, and you see difference because mostly the signals comes with the apoplastic effector proteins. So here give us an indication that the effector proteins that involved in ceratopasidiums are mostly apoplastic. And now you can see more what I mean by apoplastic. So this is a cross-section of infected cassava with a witch broom, and this is a cross-section of infected cassava with CBST and CMD, cassava brown streak disease and cassava mosaic disease. You see how the signal here, and you see how the signal there. This is exactly the difference when I say apoplastic and all, it can go inside the plant and infect the plant. So more, just to convince you more that it's apoplastic. So here you see that the first one is between the cuticle and also the plant cuticle and the epidermal cell. Or here you see this is the xylem cell, and you see how it goes on the layer between the cells, and at the end it also you see it that is moving. So I reached to my conclusion and the outlook of this. So understanding the localization pattern of pathogens in the host is critical to understand the disease. And here come how we align with this session, the pest and disease and control. So we need to know our pathogen that we can control it. And we know that the ceratopasidium colonizes the tissue and is not systemic. And we looked on the effector protein to understand the mechanism of this fungus, how it attacks the plant, and how suppress the plant. And we want to use this effector protein for later, for the control, for early resistance screening, because this you can use to also do early resistance screening and understand the defense mechanism. So I go back to cassava brown streak disease, and this is a control also aligned with our session. So this is the resistant material in the field in BRC, where we have a resistant for CBST in the field there. Thank you so much.
Thank you very much for this sort of new disease. Is that borderline?
I think I can speak up like this. I'm not sure if I understood correctly. This is the same disease that's in cacao. Is it the same fungus that affects also
Cacao?
and cacao? And if this is true, did it jump? And third question is, if this is true, what I'm saying, is it systemic in cacao and not systemic in cassava, which could be an indication that cassava is the kind of a suboptimal host for the fungus?
So we did a comparison between the genomes of the Ceratopasidium terraforme in cassava and the Ceratopasidium in cacao. There's some genes are specifically for cassava. So we don't know exactly if it jumped from cacao, but it can be a true story that it jumped from cacao to the cassava, but it adapts later because there's some genes are specific for the cassava. So it's not systemically, so because when we, as I said, it's localized. So that's why also when you have this infected stem cutting that is travel between fields and between farmer. Sometimes you saw a row and then the other row is clean, and also in the field you see it also distributed, so you don't see it continuously. So in this case, it's also proof why we don't see it like this. We don't also know exactly how is it fungus? Is the spore lay down somewhere and goes into? So until now it's not resolved how the fungus enter the plant. So if I have infected stem cutting, this stem cutting will you planted, you will get 1 out of 10. But when it's happened, how it's happened, from where it comes, until now it's not resolved. We don't know when when the sporulation happened even in the field. So this is still unanswered question. For the cacao, I don't know how I have to say I don't know how they propagate cacao. Do they propagate it by taking it out?
Yes. But I think what is really critical is, first of all, is outbreak.
Say again?
So it's an
Outbreak.
outbreak. So Sama speaks about the work that has been done in Southeast Asia. Now it has been found by our colleagues in
French Guiana.
Brazil and in French
French
Guiana.
Guiana.
In French Guiana. And there's an indication of the work that we have done by genetics, okay, that what is in French Guiana or what is in the northern Brazil, okay, has its origin in Laos. So there is some.
That's why you call it Lao 1.
Yes,
There is some transport. And I would guess, we don't know what the transport is, what the medium of transport is. But I mean, if this thing comes to Africa, then what happens? Okay, so this is really a serious outbreak, because we now see in a very, very short time, basically driven by also probably climatic aspects. Yeah, that we see this in Southeast Asia and in South America. Sorry that I.
No.
And two, okay, it's until now, because it's very difficult disease until now. It is not clear whether cacao, cassava, avocado are not the same and then jumped. So it's not clear.
What do you mean by say?
Are the same pathogen
Yeah.
and then jumped. So they come from any wild guy and then jump, and then they do a host adaptation at a later stage. So that's not clear whether a cassava one
Is
can
the
infect
same.
cacao and vice versa. So did I.
No, it's fine.
Yeah, so but this is definitely something that when we look on a worldwide scale, yeah, we have to do some sort of a, let me say, sort of a horizon scanning of what has to be on our list that can pose a threat to our plants. And this for sure is one
Yeah.
that causes serious threat.
So we have Lao isolate and we have Cambodian isolate too. So, but still need the specification will come. So until now we are not clear if they are.
And when you sequence the fungus from avocado, cacao, and cassava, it's always the same, or they are different?
They are called terrapoma. All of them called terrapoma, and terrapoma come from cacao. And this is the things, and we all the time ask ourselves, Stefan all the time criticize them, why they call it terrapoma if it's cassava? But the other names come from cacao, so all they come back to the cacao actually. The first isolate go to the cacao. So it can be all comes from cacao and it's spread.
Okay.
Any further questions to this? Thank you very much for your nice presentation. New invasions, and here we come to predatory arthropods, so that means we can get solutions. I would like to have Stella T. Tay to give you our your presentation. Thank you so much.
Thank you. Good afternoon, ladies and gentlemen. My name is Stella. On behalf of my team, I would like to present our research on abundance and species diversity of predatory arthropods in potato plants with application of botanical pesticide. The reason why we conduct the research is because potato is one of the most important horticultural product which has economic value and it can support world food diversification. As we know, after rice, corn, and wheat, potato is the fourth most consumed by people in many countries around the world. Every year, the market demand of potatoes increases, but the production rate of potatoes doesn't cope with the increasing demand. Potato farming in Indonesia involves significant chemical synthetic pesticide use due to the pest attack and plant disease. Due to high economic value, the farmer tend to overuse the chemical pesticide to keep their crop safe from pests. The thing is, the use of over pesticide chemical pesticide causes many negative impact, like such as water and air pollution, accumulation of pesticide residue, poisoning in human and livestock, killing natural enemies, pest resurgence, and increased production costs. While in agroecosystem, there are so many useful predator, useful arthropod can act as a natural enemy. Predator is one of natural enemy, can depress or can suppress the population of pests. So that's why one of alternative to decrease the population of pests is the use of botanical pesticide, which more environment friendly. So, that's why we conduct the research to compare the species diversity and abundance of predatory arthropods in potato plants treated with botanical versus synthetic insecticide. The botanical pesticide that we used is neem leaf, and Virginia tobacco stem waste. The research has conducted in Sembalun Village, East Lombok, Indonesia. Field experiment set up in potato plantation. We designed our experiment with randomized complete block design with 4 treatments. There are control, synthetic pesticide, Virginia tobacco stem waste pesticide, and neem. Sampling methods for arthropods, we used yellow pen traps and pitfall traps. From our study, we found that the predatory arthropod in our study consists of 7 orders. There are Coleoptera, Hymenoptera, Dermaptera, Diptera, Hemiptera, and Neroptera, and also Aranae. Total population of predators encountered in this study was 662 individuals, consist of 214 individuals found in neem treatment, and then 188 found in tobacco stem waste treatment, while in control treatment we found 163 individuals of predatory arthropod. Then in abamectin or chemical pesticide treatment, we found only 1997 individual arthropod. How about a species number of predatory arthropod? We found the number of species of predatory arthropods in Virginia stem waste and neem plots is highest, with 19 species each, while in abamectin we found only 17 species of predator. But in control plot, we found about 18 species, 18 number of species of predatory arthropod. So the highest diversity observed in neem, tobacco stem waste treated potato plants, and the lowest diversity found in plots treated with synthetic pesticide. From this table, we can see that the population, the highest population we found of predatory arthropod, we found in neem plots, but the difference is not significantly with with control and tobacco steam waste. But the lowest abundance we found in control treatment. If we look the diversity index, all of the treatment can be measured as medium diversity index. For the abundance index, we found that the most abundant predators were found in neem treated plots, and the lowest abundance found in synthetic pesticide treated plots. The low abundance of predator arthropod in the chemical pesticide or abamectin plots is suspected due to the predatory arthropod sensitive to synthetic pesticide. The use of synthetic insecticide decreases the diversity and abundance of predatory arthropods, while the use of botanical insecticide doesn't so much affect the diversity and abundance of predatory arthropods. Generally, synthetic chemical insecticides are highly toxic to parasitoids and predators because of their broad spectrum. Botanical pesticides are beneficial for maintaining the diversity and abundance of predatory arthropods. These predators are essential for controlling pest population naturally. Implication for sustainable farming. It is potential to reduce reliance on synthetic pesticides and promote ecological balance in potato farming. The study concludes that the botanical pesticide is most effective in maintaining the diversity and abundance of predatory arthropods in potato plants. These findings support that the idea that botanical pesticide can be viable alternative to synthetic ones, providing both ecological and economic benefit. Based on the research, it is recommended to promote the use of neem and other botanical pesticides in potato farming. This practice could enhance pest management while supporting biodiversity and reducing environmental impact. We encourage farmers, agricultural stakeholders, and policymakers to explore and implement botanical pesticide as a viable alternative to synthetic chemical in crop protection. By doing so, we can towards a more sustainable and eco-friendly agricultural future. Thank you.
Thank you very much for thanks presentation. Holding this presentation for discussion and would ask the first question. So when you compare only neem and chemical pesticides, the efficiency of neem is acknowledged by farmers. So not looking at predators, just as a pesticide, biopesticide to control any pest outbreak in potato. Is it acknowledged by the farmers? So would they easily change or do they easily adopt using neem? Because it's quite probably also a bit more expensive than using a pesticide.
The farmers know about the neem, and the neem was produced industry by industry, and we can find a neem pesticide in the store.
That's also my question. So this was a commercially available neem product that you used. This was not a locally sourced neem product, but a commercially available one.
Do you mean the neem is
Very
available?
often research with neem, this is locally produced, locally extracted. And the problem when you locally extract neem is you have a very variable concentration of azadirachtin, meaning you get very varying results in terms of control. Whereas if you buy a commercial product, the good commercial products have a defined concentration of azadirachtin A in the product, and this gives a much more reliable pest control.
Yeah. In our research, we use the commercial product of neem.
So one last question. So then I have it. When you so the biodiversity, the diversity of potatoes is clear, but do you know the contribution of those predators in a field? So it would be interesting to know how much does a predator contribute to pest control in a given field. So, you know that, or is it the question is a bit too far-fetched?
Yeah, because our study just want to know the population of predator when we use the botanical insecticide instead of chemical ones.
Okay. So thank you very much once again for this nice conversation. And here we come to our last presentation of Silvia Boletti, who is not here. She's on. She's on. Oh, sorry. So who gives us an online presentation on push-pull technology and integrated fixing. You already there? Do we have that? No.
It says.
Yes. So since she is not there, I have to thank you all for your nice presentation. So I'm predicting that she's not available, but unfortunately she's not there. So thank you very much for your nice presentations. Thank you very much everyone for his contribution in the discussion. And I can shortly summarize what we have heard today. So we see that pests and diseases play a major role, despite the fact that we often are concentrated only on few pests and diseases. If you look on a worldwide scale, from the fundamental, the new introduction, whether this be an invasive species of a plant, whether these are invasive species from Asia into Europe, whether these are invasive species into areas where it have never been an invasion before, as you see, for example, the cassava mosaic disease in Southeast Asia causing serious havoc, serious problems where we need to find very quickly solutions that we can find in certainly germplasm from either Africa or germplasm from South America. So we are facing serious threats from pests and disease, despite the fact that we don't speak much about it. Some pests and diseases are favored by climate change. So we are not only dealing with the enormous amount of movement of germplasm and planting material and things around the world, as we know. Okay, Spodoptera probably was not introduced with planting material, but rather with loads of materials from America to Africa. So it has nothing to do with planting material. So we are facing very, very serious challenges from pests and diseases that are probably aggravated by climatic change patterns, the change also of weather, so not only drought and heat stress, but just the changing weather pattern. So I think we all have to be very, very, very vigilant and attentive to the new things to come. And of course, when we look at modeling approaches, when we look at fundamental searches for the etiology of diseases, when we look at changes in pest populations, we also have to almost instantly think about management and control. So not wasting too much time with the risk assessment, but rather already looking at the things to have. Thank you very much for all your contributions. And I herewith conclude this session, thanking the audience and the speakers. Thank you so much.